Welcome biology students. I just wanted to make this quick video about our 2.5 assignment, Explain Yourself. So first thing you want to do is go to course activities. I always suggest going through the lessons because that's going to help you get all the background for this information. So we're looking at module 2, and we're looking, as we mentioned before, we're looking at the slides, patterns of inheritance. And as you can see, there are 13 slides. All of this background information is going to help you in terms of understanding hereditary patterns, how DNA creates genes, and then it looks at the idea of these genetic components that describe phenotypically what things look like, for example, the color of your eye or different things like that. And the difference between phenotype and genotype is the genetic component, the actual genes, but it doesn't always manifest the same way in terms of what you actually see, which is the phenotype. So the things can look the same, but they can have a very different genetic makeup. And then we're going through these. We start learning about Mendel, who is a great geneticist, or very much the first geneticist, who was very curious about how things are inherited, how traits are inherited. So people had some understanding that this was happening, but Mendel did all these experiments to really explore how this could happen. And so these slides go through the different experiments that Mendel did, and then further on, based on the Mendel experiments, geneticist Reginald Punnett developed the Punnett square, which you're learning about through these examples. And then as we look at slide 10, it actually explains an experiment that Mendel carried out based on his pea plants, purple and white flowered plants, starting out with he manually transferred or cross-pollinated the two plants, and then he had in the first generation they were self-pollinated, and so you could see the results of that. This is the actual assignment. So based on that first experiment with white and purple flowered pea plants, Mendel did another experiment looking at the seeds and started out with one plant that had green seeds, another plant that had yellow seeds, cross-pollinated this plant, got the first generation of yellow seeds. And then there was self-pollination here, which created that second generation here. And you go through and you answer these questions. What are the two alleles of the seed color trait being considered here? Remember, before in the information, we're going to be writing these alleles in uppercase and lowercase. Homozygous is going to either have two uppercase or two lowercase, and heterozygous is an uppercase and a lowercase. So that's dominant and recessive alleles. And then we go further in the first experiment. How does Mendel cross the pea plants? Remember, we're talking about crossing them here, and there's an explanation on slide 10 for how he does it with the purple and the white flowers. What color seeds did Mendel get in the first generation? You can see the color here. And then finally, how can you determine whether green or yellow is the dominant allele? I think it's pretty obvious as you go through this one when you have green and yellow here, and then in the progeny for the first cross-pollination, it's only yellow. You can determine that. And then what does Mendel do with the first generation of green seed plants? Okay, so these are not green seed plants. As you can see in the first generation, it's actually all yellow seed plants. So it's just worded a little awkwardly there, but the same question goes, what does Mendel do with this first generation of plants that are produced from the cross-pollination? What does he do to create this second generation? And then what is the genotype ratio of the F2 plants? And remember, genotype is not phenotype. So it's not what they look like, but what their genetic makeup is in terms of heterozygous, homozygous, all the alleles and the ratios between them. Okay, hope that helps and good luck.